Hello viewers, welcome to my channel. My name is Timothy Ekwebelem. Today, I'm going to continue from where I stopped in the topic Drug Discovery and Development Part 2. In the previous topic, Part 1, I gave a history of how drug started, which I linked to Jehovah the Most High God as the creator of medicinal plants and herbs. I quoted some biblical scriptures in Genesis chapter 1 and also Ezekiel chapter 47 on how God created the herbs and plants. And I took it from there also to talk about drug development or drug discovery in the biblical time history before Jesus Christ and even before Moses in the Bible. And from there we talked about drug discovery in before the 20th century and also the practices before the 20th century, the 20th century time and the 19th century and the recent times. In order not to bore you, I will go directly to what we have today, which I have put as 1, 2, 3, 4 up to about 16 points when we're trying to make a drug. Before you decide to produce a drug in drug discovery, you must first of all know the disease you want to find a cure for. You must have a named disease before you can even do what? Begin to choose a target. You must choose a disease. Which disease do you want or do you have in mind? Do you have cancer in mind, or do you have hypertension in mind, or do you have diabetes in mind, or HIV, or all kinds of diseases? When you have established what you want to produce, then you do what? You choose a drug target. You choose a drug target. So what are you targeting to cure? So in this one, we know what drug does in the body. When you take a drug in, some drugs were designed to bind at the enzymes to be inhibitors of enzymes and some target receptors. So we talk about receptor, enzymes, membrane. This is when we talk about where your drug targets. You target the membranes, the receptors, the enzymes, and more. So where are your drug going to target? Is it going to be an inhibitory drug? Is it going to be an agonist drug? Is it going to be an antagonist drug? Is it going to inhibit a receptor? Or is it going to be an agonist kind of drug? Or is it going to target the enzymes? There are various enzymes in the body. And if we're talking about schizophrenia now, and if you're producing a drug for schizophrenia, we know that in schizophrenia, we talk about dopamine and neurotransmitters. We know that in the theory of dopamine theory, it talks about the dripping in and the dripping out of neurotransmitters as what causes schizophrenia. So if you are to design a drug or a target drug, you have to first target the implicated neurotransmitters. And these neurotransmitters can be noradrenaline, it can be 5-HT, and a whole lot of serotonin, and these are neurotransmitters. So which of them are you targeting? So when you want to do that kind of drug, you know that you're going to find a drug that will bind at certain receptors. You know that you're targeting receptors, so you're designing a drug that will do what? That will target receptors. So sometimes you're designing a drug that will do that will target the membranes. So, and if you're targeting a drug that will that will target the membranes. You're gonna know how you're gonna deliver drugs. Some drugs are not able to do what? They are non-polar drugs. They are not polar. Sometimes when you take it in, it will not be able to do what? From the gastrointestinal tract, be able to be absorbed through the cell membrane. So if you're producing such drugs, you're going to produce a drug that will be able to do what? Survive the acidic content on the stomach and able to do what? In the gastrointestinal tract, be able to do what? Be absorbed. Be able to what? Be absorbed across by the cell membrane. Because you're trying to do what? The drug is, before it can be distributed, it has to do what? Go through drug absorption. And this absorption is through the cell membrane. So when you're producing such drugs, you're going to produce a drug that is what? That is polar. Because if you use a non-polar drugs, it will not be able to survive because of the water will be absorbed in the cell membrane. They are not, like you have lipophilicity or lipophilic drugs, are not able to do what? Will not be able to cross through the from the gastrointestinal tract to be absorbed through the cell membrane because they are not water loving. So it will not be able to pass through or be absorbed through the cell membrane. So in such a way, you're going to have to get the one that is hydrophobic, the one that love water. And such one, when it gets to the gastrointestinal tract, and it can be absorbed into by the membrane. So you're targeting the membrane. If it's the one that you're targeting that is the enzymes, some of the enzymes, you have the fat-loving enzymes. For instance, you're designing a drug that is fat-loving. Now that one drug is going to be lipophilic, and it will be absorbed from the gastrointestinal tract. It will be absorbed directly to the cell membrane, the fat cells, okay, the enzymes. So these are how you choose a target for the drug which you want to deliver to be absorbed and distributed and all that. Some drugs, because they are lipophilic, they can also be, if the drug is lipophilic or that water loving, it can easily be excreted by the liver. It can easily be excreted. So depending how you want to deliver this drug into the system will depend on how you design the drug to target the particular enzyme. So we're looking at the structure of as we're going, and I'll be able to explain when we get here. I'll be able to explain to you. So we're looking again. You have to identify the next step to identify the biopsy. So what are the test system you're using? What are the test system? Are they in vivo or in vitro? Are you going to use the in vitro? Or are you going to use the in vivo? I'm talking about using animal toxicology testing and so on. What you're going to use as your testing system. It's a testing. What you're going to use to test your drug when you have done it. Are you going to test this? You're going to use testing the animals to find out the toxicity level and everything, therapeutic level, everything, how the animal will be able to tolerate the drugs. And from there, you have to also know the identity of to identify a lead compound. So which compound are you planning to use? That's what we call combinatorial chemistry, was also where we also have compounds that has been what? That have been isolated. We'll go into the bush, you get herbs and all that and screen them, and you have isolate them and store them as a bank. So anytime the pharmaceutical company chooses to produce a particular drug for a target disease, they have to go through that lead compound to find out, to match, because it has been isolated, to match this lead compound to the name of the disease through what we call through synthetic chemistry and computational chemistry. They'll be able to design the drug to match the name target disease. So when you identify the lead compound, like I jumped it, you isolate the lead compound, purify the lead compound, and store them. Okay? So that's what I did here when I said the 12 lead component. The 12 lead component, you isolate them and purify them and store them for future use, or you can use it immediately. And the next step we're talking about is to determine the structure of the lead compound. You can see that I really jumped it. When you determine the structure of the lead compound, you know the structure of the named disease, and you can be able to do what? Design a drug. You're going to match them. There's a way to do it. You use computation chemistry, to be able to match it and check whether the drug, whether the structure has an anion, is alkyl group, you know, in a methyl group, and all that. And to depend on how you're going to use it to do the anion, the drugs that have anion also are also good. So when we're looking from there, we do what? To identify the pharmaco. Okay, so we're looking at the interaction and how we can improve the structure. We're looking at the, whether, like I just explained, whether it's the functional groups, we're looking at the functional groups, whether you have bulky functional groups or non-bulky functional groups, and how you can improve these bulky functional groups. That's where the pharmaco is. And from there, you develop and improve your target interaction. So how do you form the drug such that you will be able to look at the drug? How does the drug interact with the cells and the cell membranes and all the enzymes and all that? Then you will be able to develop your drug so that it will be able to communicate and interact and know all the binding system and what it is targeting. So we're looking at interaction with the targets, how we will modify the structure, how we know the particular functional group to put in to be able to survive a particular stage while the drug is being absorbed. So now we look at it again, we have to modify the pharmacokinetic properties. Modifying the pharmacokinetic properties. And when we talk about pharmacokinetic properties, we're talking about when the drug gets into the gastrointestinal tract, how the drug survive, what the acidity of the stomach and disintegration time, the dissolution time. We're looking at the absorption, how the drug can cross the cell membrane or blood-brain barriers, depending if it is the drug that has to do with schizophrenia or mental illness. It has to go through the blood-brain barrier, be able to pass it. And I also talk about the cell membrane, which I've already said before. These are what you consider, and before absorption and distribution. Then we're looking at what? We're looking at the half-life of the drug. Is it a long half-life or short half-life? And we're looking at how the liver metabolizes the drug. And we're looking at how the metabolism of the drug, how the liver metabolizes in the body. And we're looking at the liver enzymes. We're looking at the metabolizing enzymes. Do you have a drug, an enzyme, key enzyme that can metabolize that drug? And we're looking at the half-life and excretion. And all these things are what we look at here when we modify the pharmacokinetic. Then we can also modify through prodrugs. Prodrugs is when you mask a drug, that the drug is not able to do what? Is, for instance, you have a non-polar or a non-polar drug that does not waterlogged, and you want to pass this through this non-polar drug through the stomach. You want to pass it and you mask it with a prodrug that will be able, maybe an ester that will be able to do what? It will pass through the cell membrane, because it will mask. It's like when someone is wearing a masquerade, you don't know who's inside the masquerade because the person is wearing a mask. And until the person removes the mask, then you can know who he is, or the person that masked himself. So that is what the prodrug does. Prodrugs mask the drug, and when it gets there, and the product is able, could be an ester, will be able to do what? To be able to cross through that medium before the absorption, that cell membrane will serve it so they can be absorbed in the stomach. Once they absorb in the cell membrane, then it can now, when it gets to that place, the main drug is unmasked itself, and the main drugs will now do what? Get to the targeted receptors or enzymes. You know, that's what the prodrug does. So when you have done all this and you reach this stage, at this stage you have to do what we call patency patent. You have to patent your drug. You have to patent it, because patent is a kind of law that does what? That protects your product, that protects your drug, so that no other pharmaceutical company can do what? Will go and begin to manufacture, produce that drug and begin to sell in the market. So when you get a license from the patent body, and they grant you a license, and you will be the only sole owner of this drug when it is sold in the market. You will be the only one for a period of, in the UK, that is the United Kingdom, it's about 20 years. They give you like 20 years to have that drug. Only you will be able, only you will be the one to market this drug, your company alone. After the expiration of the 20 years, every other company can now produce a generic of that your drug. So most times, pharmaceutical companies abandon their product or their drug, because sometimes the stage of producing a drug takes sometimes about 10 years, 8 to 10, 12 years. And by the time they finish producing these drugs, they have maybe less than 8 years remaining to sell this drug. And when a pharmaceutical company look at it, they have 8 years remaining or 5 years remaining to sell. And they think that by 4 they will be able to make a profit from the drug, which they've invested over $1 billion in it. They might not be able to get their capital, talking about getting their interest, and they will abandon that. So most times they give them 20 years a time to sell, but before the pharmaceutical company be able to come to that drug, they might have spent like 8 years, and the remaining maybe 12 years is what they got to do. They will quickly be able to do what? Sell it under their own license, because they've been licensed to sell alone. And after which other pharmaceutical company can now do what? Produce that same drug. So patency protects your product. So when you patent your drug, is being protected. So in the next one, we'll talk about drug metabolism. And how does that drug metabolize? What enzymes are the key enzymes that you have in mind that you want to produce in which your drug? What is the key enzymes that will metabolize your drug to active compound or active drug? For instance, you have, I will say too much about codeine, and you have key metabolizing enzymes of codeine, which is morphine. The active principle is codeine, is morphine. So also you want to know what is the active principle and the key enzymes that will metabolize your drug, and what will be the active principle from that metabolism, and which enzymes are metabolizing your drugs. These are all the things you need to know. Then we talked about the next stage is toxicity, the toxicity test. We talked whether it's in vivo or in vitro. Now I test it in animal, we test it, and when sometimes you get a cell, if it's a bacteria, you try to put it in the cell, put the organism in it, and use your whatever injecting, and see if that the antibiotics will be able, what you produce, be able to kill it in the cell. That one is in vitro. But when you want to test it on human beings, it's called in vivo. Sorry, on animals, it's called in vivo, and that's toxic. You want to see the toxic level of the drug, whether that drug is toxic, whether it's okay. I want to check if the drug can enlarge the heart of a rat or a mice. But anything that enlarges the heart of a mice or a rat is not a good drug. So we're going to look at everything about the drug and the excretion. If when you give it to the animal, whether they are twitching their tails and whether they are feeling sleepy and all that, the only things you're checking in toxicity. Then the next plan is called scaling up into manufacturing. You make a plan on how you're going to, when you have scaled through the toxicity level and find out that the drug is not toxic, you come here, you can see here, and here plan for scaling up the manufacturing process. What is your plan? How do you design your plan? Are you going to produce it in tablet or in capsule? And how are you going to produce it? And all these things are in the plan for scaling up. And you're going to bring the new drug entity. And before you have passed through this, Then you start talking about clinical trials of the drug. When the drug has passed through the animal tests and everything, we enter into clinical trials of drug, and we talk about first trial in man. You have passed through the toxicity level, you know that it has no toxic. You start to try the first trial in man and see what this drug can do to man. That's where I've said this in several times, that's where you use healthy volunteers in phase 1, and they must get a consent, and you have to tell them what is in the protocol of the studies. You have to inform them very well, and you're going to have to allow them to really have to tell them the risk and the benefits of the trial. And you're going to tell them what the trial is all about. And you have to make sure that they've understood what you told them, and they have to go through the consent forms and understand it, after which they will do what? They can freely and willingly sign to participate. And the people that does this are healthy volunteers. And a healthy volunteer is somebody who has not got an underlying medical condition, not suffering from diabetes, not suffering from HIV, not suffering from any kind of sickness. That person, anybody that suffers from sickness, not a healthy volunteers. And in phase 1, you normally use healthy volunteers, male and female, or female, depending on what you're looking for. And like I said before, if you want to do phase 1, where you want to do a trial on antiretroviral drugs, you cannot use healthy volunteers to do it. You must use HIV patients or cancer patients or the people, because that trial will some sort of benefit them. So that you cannot try a drug of cancer drug on a healthy volunteer, or HIV drug on a healthy volunteer. So what are we looking? We're looking at the maximum response of the drug, which is we're looking at the efficacy of the drug, maximum response. We're looking at pharmacodynamics, and I say pharmacodynamics is what the drug does to the body, and pharmacology is what the body does to the drug, and a whole lot. So we'll enter into phase 2, and in phase 2 we are using patients, and in it we also determine safety parameters, because in phase 1 we did safety parameters and tolerability. Also in phase 2 we determine safety parameters and tolerability, and also using patients instead of healthy volunteers. And in this we're looking at, we do blind studies, double blind studies, where the investigator and the clinical research associate and clinical trial administrator and the physician, everybody, they will not know what the name drug is that is on trial. And I'm going to get a placebo, and I'm going to get a drug also that is existing, the replica of that drug, and we'll put it, and we'll do the trial, and after which we'll unfold the code, uncode it, and they're going to know which drugs was best that treated the disease. And also, when we have done everything here, we can enter phase 3, and where we do multi-site trials, and a whole lot of trials. So we're trying in Europe, in Africa, in Asia, and a whole lot of places. So we want to know how individuals should react to drugs, because in the way an individual reacts to drug is different from the way another individual reacts to drugs. So in this, we're checking side effects in advanced events, how the drug is metabolized, and the people that have key metabolizing enzymes of that drug. I said 1% of the entire population of the world shows absence of key metabolizing enzymes, and they show variation, and some of them show variation in DNA. So though we are created the same by God, but there's a slight show of variation on 1% of the entire population of the world. So these are what we're looking at: polymorphism of drug metabolism. How does the drug metabolize in the white people? How does the drug metabolize in black people? These are what how does the drug metabolize in Asian people? Are there side effects among African? Are there side effects among the white or the Hispanic or the Asians, and all these things you're checking? So after you have done that and you get a license from local ethics committee, you enter into phase 4, which is called the post-marketing surveillance. And here, the pharmaceutical company who has invested so much money, over $1.something billion investment in the production of this drug from scratch to finish. Now, pharmaceutical company wants to make their money, and they want to make gain because they invested, and they begin to go get approval for post-marketing surveillance of phase 4. When the local ethics committee grant them that license, like US Food and Drug Administration or European Medical Agency, what they do is they start, the drug gets into the market. And while in the market, they have doctors, they employ physicians who monitor these drugs to see if patients are suffering from side effects and all that, and they get any drug that is giving fatal drug reaction, maybe somebody died or something, drug reaction that's not good, it can be withdrawn from the market. And that's how the drug discovery and development is. That's the stages. I've just explained everything, and that comes to the end of the lectures. And I will advise you to subscribe to my channel, Timothy Ekwebelem. I also advise you to click at the bell on the screen. Thank you very much for watching and listening. God bless you. Bye.